Welcome back, and indeed to Lecture 5. Let's continue our exploration of biology, shall we? The characteristics of an individual have a bearing on its survival, on its chances to reproduce, and on the success of its offspring. Such factors drive evolution, as different alleles undulate in populations. Genetic variation, that is, differences in alleles of genes found within individuals of a population, provide the raw material for natural selection. Natural populations contain a wealth of such variation. In this lecture, we explore genetic variation in natural populations and consider the evolutionary forces that cause allele frequencies in natural populations to change. Through time, species accumulate differences. As a result, descendants differ from their ancestors. In this way, new species arise from existing ones. Natural selection produces evolutionary change when some individuals in a population possess certain inherited characteristics and then produce more offspring than individuals lacking these characteristics. As a result, the population comes to include increasingly more individuals with the advantageous characteristics. A rival theory championed by Jean-Baptiste Lamarck was that evolution occurred by the inheritance of acquired characteristics. According to Lamarck, changes that individuals acquired during their lives were passed on to their offspring. Lamarck proposed that ancestral giraffes with short necks tended to stretch their necks to feed on tree leaves, and this extension of the neck was passed on to subsequent generations, leading to the long-necked giraffe. In Darwin's theory, by contrast, the variation is not created by experience, but is the result of pre-existing genetic differences among individuals. Natural selection, by favoring individuals with certain alleles, can lead to change in such allele frequencies. But it is not the only process that can do so. Allele frequencies can also change when mutations occur repeatedly, changing one allele to another, and when migrants bring alleles into a population. Evolution can result from any process that causes a change in the genetic composition of a population. We cannot talk about evolution, therefore, without also considering population genetics, the study of the properties of genes in populations. Biologists have always wanted to know how much genetic variation exists in populations. Initial approaches to understanding variation examined the most obvious differences, the morphological. At this level, natural populations usually show substantial genetic variation, e.g. polymorphic variation, as Figure 4 illustrates. Two forms of genetic polymorphism exist: transient polymorphism, in which a particular form is in the process of spreading through a population, causing the relative proportions of phenotypes to alter, and balanced polymorphism, in which two or more forms coexist in a stable ratio within a population, each form possessing both advantageous and disadvantageous characteristics. Genetic variation within populations was a puzzle to Darwin in the mid-1800s. Meiosis, a type of nuclear division that gives rise to 4 reproductive cells, or gametes, each with half the chromosome number of the parent cell, had not yet been discovered. The theory of blending inheritance, in which offspring were expected to be phenotypically intermediate relative to their parents, was widely accepted. If blending inheritance were correct, then the effect of any new genetic variant would quickly be diluted to the point of disappearance in subsequent generations. Godfrey H. Hardy, an English mathematician, and Wilhelm Weinberg, a German physician, independently came to the conclusion that the original proportions of the genotypes in a population will remain constant from generation to generation, as long as the following assumptions are met. Consider a population of 1,000 cats in which 840 are black and 160 are white. The frequencies of the two phenotypes would be 0.84 or 84% black, and 0.16 or 16% white. If we assume that the white cats are homozygous recessive for an allele we designate as lowercase b, and the black cats are either homozygous dominant, that's BB, both uppercase, or heterozygous upper and lowercase b, we can calculate the allele frequencies of the two alleles in the population from the proportion of black and white individuals. Let the letter P, that's lowercase p, designate the frequency of the uppercase B allele, and the letter Q, the frequency of the alternative allele. Because they are only two alleles, P plus Q must always equal 1, that is the total population. In addition, we know that the sum of three genotype frequencies must also equal 1. If the frequency of the uppercase allele is P, then the probability that an individual will have two uppercase B alleles is simply the probability that each of its alleles is an uppercase B. The probability of two events happening independently is the product of the probability of each event. In this case, the probability that the individual received an uppercase B allele from its father is P, and the probability the individual received an uppercase B allele from its mother is also P. So the probability that both happened is P times P equals P squared. By the same reasoning, the probability that an individual will have two lowercase b alleles is Q squared. The individual could receive a B uppercase from its father and a B lowercase from its mother, or vice versa. The probability of the first case is P times Q, and the probability of the second is Q times P. Because the result is, in either case, is that the individual is heterozygote, the probability of that outcome is the sum of the two probabilities, or 2PQ. In the next generation, the chance of combining two uppercase B alleles is P squared, or 0.36, that is 0.6 times 0.6. And approximately 36% of the individuals in the population will continue to have two uppercase B genotype. The frequency of BB lowercase individuals is Q squared, 0.4 times 0.4, and so will continue to be about 16%. And the frequency of BB upper and lowercase individuals will be 2PQ, 2 times 0.6 times 0.4, or an average 48%. Darwin proposed that evolution of species occurs by the process of natural selection. Other processes can also lead to evolutionary change. For a population to be able to evolve, it must contain genetic variation. DNA testing shows that natural populations generally have substantial variation. To reiterate, evolution can be described as descent with modification. Natural selection occurs when individuals carrying certain alleles leave more surviving offspring than those without the alleles. Natural populations generally contain considerable amounts of genetic variation. Population genetics studies this variability through statistical analysis. The Hardy-Weinberg principle states that in a large population with no selection and random mating, the proportion of alleles does not change through the generations. Finding that a population is not in Hardy-Weinberg equilibrium indicates that one or more evolutionary agents are operating. I welcome you back to Lecture 6 when we discuss these evolutionary agents. Goodbye for now.